New chapter. Shh, guys, new chapter. New beginning. Okay? Chapter 7. This is a chapter. This is a chapter that is filled, as you can see from your vocab list, with a lot of big words. But it's actually, I think, one of the more fun chapters to learn about because you're learning about all the parts of the cell. Okay, and these are words that you've seen a lot of those words before from past science courses, mitochondria, plasma membrane, those kind of deals. So I think it can be a potentially easy chapter. Before we talk about the parts of a cell, let's talk about microscopes for a minute. Tomorrow we're going to be most likely doing a lab, depending on if you can prepare for it and whether you can behave in it. So, we're going to have to kind of get a little introduction to the microscope. Now, I know some of you guys work with microscopes back down in junior high, but some of you did not. So I'm basically kind of treating this like no one's ever used a microscope before. So another thing you're going to have to do for this test is learn the parts of a microscope. And I will show you that picture here in just a minute. Before I do that, a little introduction about what microscopes are. There's two major types of microscopes, a light microscope and an electron microscope. The microscopes that you guys have ever seen are light microscopes. And they're called light microscopes because, well, they operate by a light that's at the bottom that shines up on the specimen so you can see it. Now, the nice thing about light microscopes is you are able to view a living or a non-living sample using light microscopes. Now, the electron microscope has a big disadvantage. You can't look at anything living under an electron microscope. Tomorrow we're going to be doing a lab. I'm going to go out to the Reservoir Van Wert tonight and get some pond scum, basically some pond water. And you guys are going to be able to look under the microscope tomorrow and find all these little critters in that sample. It's a pretty cool lab, actually. You'll see things from the plant kingdom, things from the animal kingdom, things from the protist kingdom, probably even things maybe from the fungus kingdom. Okay, anyway, you'll find a ton of stuff floating around. And the nice thing about light microscopes is you can view that stuff while it's living. You'll see cells actually feeding on little pieces of algae kind of deal. It's really neat. It's really neat. It's a neat advantage of a light microscope. Electron microscopes, you may not even ever see one in your lifetime. They're huge. They basically fill up half of a room, and you basically, in order to use one, you sit at a computer desk. So they operate with a computer screen. Your sample's somewhere over here. These work by shooting like beams of electrons into your sample, which are able to generate this picture that you see on a computer screen. Very cool. And what the advantages of these, as you can figure it out, extremely high resolution. Resolution means, clarity. These things are so sharp. These images are awesome. Very, very, very cool. However, you can't look at a little critter floating around using one of those things. So that's the only bad thing. You have to actually, to prepare your sample for an electron microscope, you got to kill it, treat it with some chemicals, and what? The only way that you just can't, they have to, when they are passing beams of electrons through this thing, it's got to be prepared with a special chemical, and any chemical is going to just take out a living organism. So, advantages, disadvantages. The microscopes we have here are not just light microscopes, they're actually called compound light microscopes. What does the word compound mean? Compound. What if you get a compound fracture?
Made up of two or more.
You got it. That's exactly what it means, okay? You got more than one. Very good. So a compound light microscope is called a compound light microscope because it has two lenses. Okay, here's what I mean by that. In your compound microscope, two lenses. There's actually one up here, believe it or not. That's the one I'm talking about. The other lens is located inside one of these objectives. These things are objectives. They're called objectives, and inside of the objectives are lenses. So one lens here, one lens here. That's why it's called a compound microscope, because it's more than one lens. Where is that picture of the compound microscope? Here are the parts of a microscope you need to know.
Holy crap.
I actually have a copy for each of you of this.
Don't
Don't draw that out. Would I be so mean? Yeah, I would.
So do you really want me to answer that question? Or?
Oh, you know what? I gotta take
How was your better one? Mine was boring.
Okay. Oh, I already got you.
She does.
All right, guys. So basically, you're staring at the same thing that's up on the board. These are the parts of a microscope that I want you to be familiar with. Your test will actually have a whole separate page, and I'll have a picture of a microscope, and you'll have to fill in the blanks of what all these parts are, kind of deal. Okay. Okay. Ocular lens. That's the one lens I was telling you about. It's also an eyepiece. Shh. By the way, there's a built-in pointer. Tomorrow when you guys look through these things, here's what ends up happening. You look through them, and you're like, what's this like big black line running through my specimen? Well, that's a pointer. That's meant for pointing things out to your neighbor. That's cool. Like, hey, I have the pointer on this really cool thing. Look at it. Don't confuse that big black line going through it with something that shouldn't be there. It's actually meant to be there. Body tube, nose piece. Okay, this nose piece is basically, you have these objectives hooked up to this nose piece. This particular microscope has 4 objectives. The shorter the objective is, like the one that's in place right now, the shorter it is, the lower the magnification. So that's low power, in other words. When you start and end an experiment using microscopes, you will always want to put the low power in place. That's one of Mrs. Hatcher's pet peeves, is when the low power is not in place. You never, for example, want to leave the microscope like that and walk away at the end of the day. Okay? No. Hey, another thing about these objectives, when you just learn how to use a microscope, one of the first things is, I can't find anything. I can't see anything. Let me tell you what 8 times out of 10 the problem is. These objectives have to click into place. Listen, you probably didn't hear it, but they have to actually click and lock into place. Sometimes I'll come over to a table, I can't see anything. Well, yeah, because your objective is not locked into place. So always check that, okay? Seen that happen a million times. Okay, moving along. So, by the way, these objectives, these are like the expensive part of the microscope. That's where all the cost comes from. These things are about $400, $500 each, okay? This is why it's extremely important when you're working with a microscope that you go slow, because these things are expensive. The reason these things get damaged is sometimes people are on like high power. They're using maybe one of the longer objectives, and they're not being careful when they're focusing, and they're moving this stage up and down, and the objective comes in contact with the light source, and the 2 pieces of glass smash each other. That ruins the microscope. That's $500 gone. The point is, when you're using a microscope, be very aware of which objective is in place, and be sure when you're focusing that piece of glass and that piece of glass are nowhere near each other. Okay? You just got to be real careful with these things. Other pieces, parts. Oh, you can, yeah, you guys have a sheet, so I won't. That's the diaphragm. I want to see the diaphragm. Okay. This is another reason people have trouble finding things, because this diaphragm is literally this little silver lever that's underneath. You can barely see it. Play with it though tomorrow. A diaphragm is just like a fancy word for a light filter. Depending on what you're looking at, you may need more light to see that thing, or you may need less light to see that thing. And that basically is the lever that controls how much light actually comes through from the bottom. So that's kind of important. That's another reason sometimes these things get flipped all in one direction and it makes everything black when you see it. Just got to make sure that you can adjust that. This black thing is this stage, okay? You place your microscope slide. Always use these things with the microscope slide. And basically you'll put that in place, lock it into place, and there's a bunch of levers here. You have this coarse focus, which is right here, and you have this fine focus, which is right down here. When you're just going up to your microscope, looking through, you'll want to use your coarse focus to basically find something. You'll want to use your fine focus to fine tune it, to basically make it clear. So tomorrow, you use a combination of your coarse focus and fine focus to kind of find your organism. Up, down, side. Okay, I don't even know what those refer to these little levers right here. You're going to have a microscope slide in place. You will use these little levers to move the microscope slide around so you can look for critters. You'll use these a lot tomorrow, because your little critters are going to be floating and swimming and this and that, and you're going to have the hardest time trying to follow them. So you won't want to use your fingers and move the slide all around. That's pointless. You'll want to use these two levers over here to move the slide around so you can find your organism. Yeah, these little critters are fast. It's a challenge to view them. So I think that's basically it. I know until you have a microscope in front of you, it's hard to actually learn about it. Couple golden rules, though. This is what I wanted to tell you. When you guys hold a microscope, always with two hands. One at the bottom and one around the arm. Okay, one around the arm. Because what I'll have you guys do is the microscopes are in the middle room. You'll have to bring them out. The other golden rule, when you begin and end an experiment, always on low power, the short objective. Another rule, there's this one objective here that has the word oil on it. You'll never use that. The only way you use that objective is when you have to use it with a sample of oil. We won't do that. Okay, so never, ever, ever, ever will you ever use the objective of this thing, and you can even see the word oil on the side. So there's kind of your introduction to a microscope. Yeah, you guys, I get real worried about equipment staying in good shape. And microscopes are one of the more important things we have in the lab. So it could be a really great lab. You'll be able to see some really cool critters, but let's make sure we just kind of treat that equipment with love and care, because it's a good idea to do that. Okay. Moving along. Oops. Okay. Okay, surface area to volume ratio. Eesh. Whoa, sounds like math class. Okay. Kind of in order to teach this point, I'm going to really, really simplify it. Let's say I have a box. You got to imagine this thing's like 3D, but I'm not going to draw it 3D. And let's say I have right next to it, I have 4 smaller boxes. But the 4 smaller boxes, oops, they're not the best. You guys I know can draw better than I can. The 4 smaller boxes are actually going to occupy the same volume as the other one. In other words, they take up the same amount of space. The box on the left, the box on the right, they take up the same amount of space. The only difference is the one is broken up into 4 pieces. Okay, you could, if you want it, you could compare this to like a sugar cube. And if you took that sugar cube and kind of chopped it into 4 pieces, there you go. You got your sugar cube in 4 pieces. You're like, wait a minute, isn't this science class and not math class? Yes, there's a point here. Cells. Remember, cells are the basic unit of life. Cells need a high surface area to volume ratio. Okay, here's what that means. Surface area to volume ratio. Ratio is just a comparison number. In other words, if you're comparing surface area and volume of 2 things, you want cells to have a high surface area to volume ratio. What does that mean? You want cells to have a lot of surface area. One of you guys could probably tell me this. Let's say I have 2 cells here. Okay, they're represented by models here. I have 2 cells, a big cell and a little cell. Imagine that there was a little carbon dioxide molecule. Hey guys, remember what carbon dioxide is? Carbon dioxide, I'll just write it off to the side. Carbon dioxide is that puny little molecule. It's a carbon with 2 oxygens attached to it. Teeny tiny. Okay. Anyway, what you guys are going to learn in the future is that when your cells are actually little factories, and one of the waste products they make is CO2. If CO2 accumulates in your cells, you're in trouble. That cell's going to die. CO2 is toxic. Carbon dioxide is toxic to cells. So here's my point. Imagine I have a CO2 molecule in each of these cells, and it's smacked right in the middle of them. It's in the center of this one, and the center of this one. Imagine you're that CO2 molecule. If you want to get out, which cell would you move out of faster? Which cell would you have to swim through less to get to the outside? This one. Okay, that's good for the CO2. So if I were a CO2 molecule, I would rather be in this thing than in this thing because I have a better shot of getting to the outside world in this thing than I do in this thing. This goes along, this has a high surface area to volume ratio. It's the small volume and there's a lot of surface area. This one has a lot of surface area, you would think, but it's also so darn big. Again, that CO2 molecule is going to have to it won't get out very easy. One thing you guys are going to learn, if your cells inside of you right now, all those trillions and trillions of cells that are making you work right now, basically, they need to move stuff in and out quickly. They don't have time to swim from here to here to get out. My point is cells better be small. Your cells better be small. Let me look at it a different way. You're going to be looking at a bunch of cells tomorrow under a microscope. And these critters are cool. And they're living organisms, but they're 1 cell big. Okay. One of them is called an amoeba. All right. Let's imagine that for just a minute that suddenly that amoeba that you guys are going to be looking at tomorrow grew to be the same size as you are. It didn't increase in number of cells, just that 1 cell just went boom and got really big. That amoeba wouldn't last long. It would not last long at all. Because basically, since every cell is going to be making waste products, those waste products need to get out, that cell's not going to be capable of moving that stuff in and out very easily. So, for some reason, they want you to know what surface area to volume ratio is. I think it's just easier to kind of treat it like I did, of just thinking of living organisms. Living organisms, the bigger we are, the bigger a living organism is, the more cells it better be made out of. So, cells need to be small in order to move substances in and out quickly. Very important. Speaking of which, just to kind of make it more realistic, let's say I have a cell. I already told you CO2 really needs to get out of cells quickly as soon as it's being made. Can anybody think of anything that needs to go into a cell? What do you think your cells need a lot of in order to work properly? Not all cells, but many cells.
Sugar.
Good. Glucose or sugar. Very good. That's one of those things that needs to move in quickly. Good job, Kenny. Another thing that your guys' cells need a constant supply of quickly, what's that? Oxygen. This is why you guys have to breathe. You breathe so every one of the cells inside of you can get some oxygen. What else we move in and out of us? Kind of goes to both sides. Water. We're going to be learning in this chapter, actually, that water really kind of goes back and forth in and out of a cell. Why? Because your cells are constantly changing environments, and the water needs to kind of basically come in and come out depending on where it's at. You don't want your cells exploding, do you? And you definitely don't want your cells shriveling up. So that water kind of comes in and out to maintain that healthy cell-like appearance kind of deal. All right, enough on that. Almost getting there, guys. I know you love these long classes.
Oh, yeah, I love them.
Now let's get to the fun part. Cells and stuff inside of cells.
Are you lying to us?
Nah, it's all good. I don't know. I want to know more about the kind of things that make up my insides. All living cells. I don't care if you're a bacteria, if you're a fungus, if you're a plant, if you're an animal, doesn't care. All cells fall into one of two categories. They're either considered a prokaryote or a eukaryote. Do you guys remember those words at all? You should. You should have seen them before. We need to put that into our memory banks. There are either prokaryotes or eukaryotes. I always remember prokaryotes being the simple cells. They are. Because if a prokaryotic cell, prokaryote, it doesn't have a whole lot to it. Very simple. We'll go into details here in a minute. Also, the big difference is that all those cells that are prokaryotic, they don't have a nucleus. What's inside of a nucleus? Anybody remember? The nucleus holds something really important on the inside. What is it? Mrs. Hatcher's favorite molecule? It's painted on the back wall.
DNA.
Yeah, good. Okay.
Nerd.
Hey, a nucleus is like a container for DNA. Prokaryotes don't have it. Now, they have DNA. Don't get me wrong, but the DNA in a prokaryote just kind of wanders around without being enclosed in a nucleus. I'll show you a picture of one in a minute. Eukaryotic cells have a nucleus, and their DNA is inside of that nucleus. Question for you. There are 6 kingdoms of life. Name me whatever you know.
Plant, animal.
Insect.
Plant, animal,
Insect.
fungus. Insects are part of the animal kingdom. Bacteria actually has 2. So there are 2 that make up the bacteria. What are we missing? So we have plant, we have animal, we have fungus. We have the 2 ones for bacteria. We're missing one. Give you a hint. They kind of grow up in your yard when you're after
Grass.
it rains for a
Weed.
really
Mushroom.
long time.
Moss.
What's a mushroom an example of?
Weed.
Fungus. Don't forget about the fungus. Okay?
Say what?
Good. Actually, guys, you did pretty good. Hey, those are those 6 kingdoms we just talked about. Hey, look at it.
Yeah.
She counted practices.
Eubacteria and Archaebacteria are the 2 kingdoms that just have bacteria in them. Man, for being so small, they occupy 2 of the 6 kingdoms, and their words are the biggest. That's what's so ironic. You have these big, giant words to represent really tiny things. But I'll tell you what, though. Those things are awesome, strong little suckers.
Like me.
Okay. Fungi, protist, plant, animal. Basically, the 2 kingdoms that have the bacteria in them, those bacteria are prokaryotes. Bacteria are like the simplest life forms out there. Now, don't underestimate them, though. Just because they're the simplest doesn't mean that they're not. Well, think about it. Just because they're the smallest and the simplest doesn't mean that they're any less of a threat. Think about almost every illness out there are pretty much bacterial infections. Those little suckers have an amazing ability to do harm for as small and simple as they are. But they're simple. They don't really have a whole lot in them. Fungi, protists, plants, and animals. Oh, by the way, protists, that's the one we were forgetting. That's what a lot of the things you're going to be looking at in the microscope tomorrow are protists. Usually protists are one-celled creatures. They're not bacteria. They're different. They're like one-celled organisms. Let me show you a picture of a prokaryote.
Wow. It's like a broom.
That's a bacteria. That's like a typical bacteria. Not a whole lot to it. Okay, that thing has some DNA. Okay, it does have DNA, but it's not enclosed in any special container. It's just kind of all over the place. And you have a cell membrane. This particular bacteria has a cell wall. They all don't. Not all of them do. And they have these ribosomes. Okay. One more thing to write down, and then I'll kind of be done here. And I'll show you some more pictures, actually. The last thing I want to talk about are what all cells have in common. I don't care if you're a bacteria, or if you're a cell that makes up a fungi, or a cell that makes up a protist or plant or animal. I don't care what kind of cell. If you are any living cells, you are going to have all of these things to you. You're going to have a membrane, plasma membrane. You're going to have something called cytoplasm, which is just like the fluid portion inside the cell. Every cell has one of those, has that. Every cell, don't care what it is, are going to have ribosomes. And every cell is going to have DNA. Remember, one of those characteristics of life was having genetic material. Well, every cell's got to have some DNA. It doesn't necessarily have to be enclosed in a nucleus, but you got to have DNA. I think I already showed you a picture of a prokaryote. Let me show you. I know I have a picture up here of your typical eukaryotic cell. If I can find it.
Did you just show us the prokaryote? Did you show us that?
Yeah, I did just show you that. It's not the best picture. I wish I had a better one. And don't look at what's off to the side. Just look at that thing right there. This is like your typical eukaryotic cell, and they didn't label anything, but just this is the only thing I wanted you to see. There's a lot more stuff inside. There's not just ribosomes. You've got these purple things. You've got this ribbon-like structure. You have this nucleus right here. You have other things going on. In other words, eukaryotic cells are much, much more complex. They have a lot more to them. Hold on, a couple other pictures. What do you want, Kyle?
There's some like thing in the plants that's not. Like I remember from Mrs. Fisher's.
Does anybody know?
She stressed that.
Cells, yeah, no, that's important. We'll go over all of that again. Who could tell me the difference between it's a cell wall? Very good. Anybody else know the difference between a plant and animal cell?
Wow.
Yeah, chlorophyll. and chloroplasts. Yep, very good. Good, and a cell wall. And plant cells actually have one other difference between them and animals. Think about a plant.
I don't have to sell those.
Plant, they do. They do. Listen, plants still have a skeleton. So, as part of their cells, they actually have this big, a real big organelle inside of them called a vacuole that holds water. In other words, plant cells have this big, giant storage tank of water. Animal cells have one, a vacuole, but it's not nearly as big. We'll go over those details. That was good, Tyler. All right. I like this picture. Now, you're looking at a eukaryotic cell here, definitely. But I like this picture because it's basically, and we're going to go into detail about all these parts of a cell. Yeah, these are people. Please understand there's not real people inside your cell. Okay?
I know, I'm just saying.
I know, but that's why I hesitate to show these pictures. Please don't think there's little forklifts inside your cells. But I like this picture because I want you guys to understand that inside your cells, of all living organisms, a blade of grass or inside of you or a bacteria cell, I don't care what it is, there's a whole lot going on. There are a lot of parts. Now, bacteria don't have as many parts. There's a lot of parts. Each of the parts has a job. And they kind of, in this picture, they're showing you that it kind of operates like a factory, where in a factory you have like a shipping and distribution center, you have a manufacturing area where you're making your product, you have this, a storage facility. That's what they're showing you about a cell here, and that's exactly what happens. Look here's my storage facility. Looks like you have boxes over here. That's the vacuole, by the way. This purple thing is your nucleus. Inside of it is a piece of DNA, although you really can't see that. You have these ribbon-like structures that are, you see the little guys like carrying something over their shoulder and moving along. They're kind of like little, like a highway system is what I call it.
For those things.
Huh?
So I'd use a forklift for those things.
Yeah, well, the little forklifts are actually supposed to represent these little things called vesicles, which they kind of move things as well. They're kind of little movers. That green thing, that's a chloroplast. Okay, not all cells have that, only plants. Anybody know what that thing's supposed to be?
I know I can think of it.
They have a little pole next to it with a little light bulb, so it makes energy. What's the organelle that makes energy? Starts with M. Mitochondrion. Okay, good. So over the next couple of days, we're going to kind of be talking about each of these organelles. It's actually kind of fun, guys, just to understand that in every one of the cells in your body, there's a lot going on. We're going to be talking about why your cells are called factories. What are they actually making? Remember what we are? We are big bags of protein. Well, there's the factory right here. It's making your proteins and your lipids and your carbohydrates. All of that's being made in the cell. So we're going to be talking about all those pieces of parts in the next couple of days. Any questions?
No.
All right, we're done.
No.
Should we think for any questions?
Questions?
I don't want to get